Good evening, everyone. It's already more than 6:15, and we'll now start our session. This is week 5, and we have already completed 4 weeks of our live interactive session for the animal physiology course. And I welcome you all. I'm not sure how many new students are here, and I can see some old names. So I'm hoping that everyone is attending the class on a regular basis. In the previous two sessions, we have discussed about muscles and a little bit about neurons and nerves. And today's session will be all about nerves and membrane potential, plasma membrane potential. Okay, and I'm hoping that everyone has. Okay, there was some network issue. I hope I'm audible to everyone. Isn't it? Okay, so the rule is same that we will be discussing 10 questions today, and this will help you in solving the present year questions as well. And I'm hoping that everyone has attended the course lecture and then they have joined this interactive session. Then only it will be useful to you, right? This is like a practice session of what you have learned through the lectures of the week 5. So the rule is same. I'll read out the question. You can see the question on the screen, and I'll give you 1 minute time, and you have to answer the questions in the chat box, and then after that we'll discuss the correct answer and a good explanation to it. Okay. So I'll start with the first question. Okay. So the first question is, which of these following ions govern the resting membrane potential in neuronal cells? Which of these following ions govern the resting membrane potential in neuronal cells? Na plus K plus, Na plus K minus, K plus Na minus, or N plus K plus? Your time starts now. Start answering. Okay, this is a very easy questions, and I'm hoping that everyone will give the correct answer. And yes, of course, I want every one of you to note down how many correct answers and how many wrong answers you're giving, and we'll see what score you have scored in the end of the session. Okay, and everyone, please be active and try to answer. Even if you are not sure about the answer, try to answer it in the chat box. Be active. Okay, most of you are saying A. Okay, let's see if A is the correct answer. Yes, A is the correct answer. So, first of all, what is resting membrane potential? Resting, as the name says, the membrane is in a relaxed state. And what happens when the membranes are in a relaxed state? Obviously, the charge of the membrane is all about the movement of the Na plus and K plus ions and the concentration of ions outside and inside of the membrane. the concentration of K plus and Na plus ions inside the cell, that is in the cytoplasm and in the extracellular fluid, is what governs the charge of the membrane. Is it at a resting potential, like the action potential of the membrane? Is it depolarized or repolarized? We'll discuss all these terms one by one in the following questions. So what happens that at the resting potential, everything is at rest. There are voltage gated channels, that are voltage gated sodium channels and voltage gated potassium channels, as well as ATPase pump. All of these are at rest in the resting potential, and at the resting potential the cells are at a potential of minus 70 millivolts, right? And the Na plus K plus transporter pumps K plus ions into the cell and Na plus ions out. This is the function of the Na plus and the K plus transporter present here, and the ATPase pump, we'll discuss about that later. So for the simple question was, what are the membranes involved in the resting potential? So that is Na plus and K plus. Okay, we'll move on to the next question. Which of the ions has higher extracellular concentration? Which of the ions has higher extracellular concentration? K plus, Cl plus, Na plus or N plus? Your time starts now. Higher extracellular concentration. So which of the ions are present in the outside of the cell? That you have to answer. Okay. Think wisely and then answer. Whoever are joining late, try to cope up with the questions. We are in our question number 2. But please try to join in time. It's already 6:25 and people are still joining. Someone is presenting their stream. Please switch it off. Abdul Salim Kill. I guess you have switched on your presentation. Okay. Abdul Salim, whoever is it, please stop presenting your screen if you can hear me. It's creating a disturbance in the class. Okay, I guess everyone has answered. So we'll discuss. So the correct answer is Na plus. I am seeing variable answers, but the correct answer is Na plus. So whoever has given Na plus as the correct answer, good. So Na plus concentration is higher in the outside of the cell, and K plus concentration is higher in the inside of the cell. As you can see here, this is the outside of the cell and this is the inside of the cell, and these dark blue color ions are the Na plus ions. These are present in more number. The concentration of Na plus ions is more outside of the cell. This is in a resting condition we are talking about, right? In a resting condition, that is when the cell membrane is at resting potential. Okay, and in the inside of the cell, the K plus ions are more these green colored ions you can see here. Okay, next we'll move on to the third question. In equivalent circuit, cell membrane is shown as only capacitor, only resistor, both A and B, or none of the above. There is a circuit model of the cell membrane. If you have attended the lecture, you must have known. In equivalent circuit, cell membrane is shown as only capacitor, only resistor, both A and B, or none of the above. The time starts now. Start answering. Everyone, please try to answer. The cell membrane is shown as the cell membrane. Okay, read the question wisely. Okay, everyone is saying A. Someone has said C. Nirmita. Okay, everyone, please mute your audios and. Okay, okay. I guess everyone has answered. So the correct answer is C. That is both A and B. I hope I am thinking that most of you have not attended the lectures. Only one girl could give the correct answer. Okay, we'll discuss. So, this is the model of a cell membrane which we generally study, and this is the circuit model of a cell membrane which is equivalent to this, but it is made in the form of a circuit with resistors and batteries and capacitors, right? These are the batteries and resistors and capacitors symbols we learn in physics. Okay, so what happens? This is the circuit model of a cell membrane which is equivalent to this model of cell membrane, that is K plus channels, Na plus channels, and chlorine channels. The same, it is occurring as a resistor in the circuit model, that is the Rk is the K plus channels, potassium channel, sodium channel, and chlorine channel. So these are acting as resistors, right? The variable resistance represents gated channel conductance, that is the gated channels. Batteries represent the driving force of ions, the force through which ions are moving throughout the membrane. This is the across the membrane. This is the battery, and the capacitor represents the insulating property of the membrane lipid bilayer, right? The capacitor represents the insulating property of the lipid bilayer. Because of its insulating property only, the ions are able to move across the membrane, right? So it, the cell membrane has all the things, that is the resistor, the capacitor, the batteries. So our question was the equivalence membrane. The cell membrane is shown as both capacitor and resistor. Most of you answered only capacitor. That is a wrong answer. I hope you have learned from this. We'll move on to the next question. Which of the following rebalances concentration of ions to resting membrane concentration after inward sodium current and outward potassium current? Read the question carefully and then answer. Which of the following rebalances concentration of ions to resting membrane concentrations after inward sodium current and outward potassium current? The time starts now. Start answering. Think properly, read the question properly, and then answer. Some are giving answer A, some are giving D. Is everyone done? Is everyone answering? I want every one of you, whoever is present, to answer. Read the question properly and then answer. Have a lot of time. Okay, is everyone done? Okay, okay. We'll see what is the correct answer. So the correct answer is D. Most of you have given the correct answer, I guess. ATPase pump is the one which rebalances the concentration of ions throughout the across the membrane. So as I have told before, the concentration of Na plus ions is more in the extracellular matrix, that is in the extracellular fluid of the membrane, and K plus ion concentration is more inside the cell. So after a certain time, when the concentration is at the highest level, that is the extracellular fluid has the highest concentration of Na plus ions and the cytoplasm has the highest concentration of K plus ions, at that time this ATPase comes into action, that it exchanges the ions through this pump. That is, 3 Na plus ions go out, for 2 K plus ions come in. This is the ATPase pump which rebalances the concentration so that the cell can go again to the resting membrane potential. So when the concentration of ions, that is the sodium ions in the extracellular fluid, and the concentration of K plus ions in the cytoplasm is highest, that is something else. That is the depolarization and repolarization, which we'll discuss in the further questions. But for this, the only question was, which is the pump which rebalances the concentration of ions across the membrane. So it is the ATPase pump, which through the release of ATP sends out 3 Na plus ions outside and helps enter the 2 K plus ions into the cell. Okay. So through this, the Na plus concentration in the extracellular fluid and K plus concentration in the cytoplasm is balanced. Okay, we'll move on to the next question. Nodes of Ranvier are myelinated zones, non-myelinated zones, are on dendrites, or none of the above. This is a very easy question. Nodes of Ranvier are myelinated zones, non-myelinated zones, are on dendrites, or none of the above. Okay, most people are saying A. Some have said B. Okay, we'll see which is the correct answer. So the correct answer is B. Nodes of Ranvier are non-myelinated zones. If you doesn't understand the thing this properly, you will be confused, because nodes of Ranvier are present in myelinated neurons, but they are not myelinated. They are present in the gaps between these myelin sheaths in the new axon of the neuron. So here you can see the neuron structure. You have the cell body, you have the dendrite, this is the axon, this is the terminal end, and the axon is covered by this myelin sheath. When this is covered, we call it as a myelinated neuron, and if the myelin sheath is absent, we call it as unmyelinated neurons. So nodes of Ranvier are present in the gaps between these myelin sheaths, which are unmyelinated, right, or non-myelinated regions of the neuron. Okay, we'll move on to the next question. Myelination in peripheral neurons is done by. Myelination in peripheral neurons is done by Schwann cells, astrocytes, microglia, or oligodendrocytes. Myelination in peripheral neurons is done by. Your time starts now. Start answering. So there are two types of neurons, right? One is peripheral neurons and the other is, who can say? Everyone try to answer the question. I'm not seeing the equal number of answers as the students present in this class. What is the other one? One is peripheral neurons, another one is, no one knows. I'm requesting everyone to please attend the lectures. That will only help you, that will only serve the purpose of joining this course, right? I'm hoping everyone is able to understand the lectures and through this class they're able to practice the concepts they have learned in the lectures. Okay, and you will get few more extra questions in the present assignments. So the practice will be good. Some are saying central neurons, sensory neurons. Okay, we'll see. So we'll see what is the correct answer. Correct answer is Schwann cells. Congratulations, most of you have given the right answer, I guess. So here you can see there are two systems. One is the peripheral nervous system and the central nervous system in our brain and in our nervous system, if we say, and the peripheral neurons are present in the peripheral nervous system and the central neurons are present in the central, or sensory neurons are present in the central nervous system. So the myelin sheath in the peripheral neurons are made by the Schwann cells and the myelin sheath in the central nervous system neurons are made by the oligodendrocytes. Okay, there is a difference between the two. Both of them, both the myelinated neurons in the both the systems have nodes of Ranvier, and as the question says, myelination is done by Schwann cells in the peripheral neurons, whereas myelination in the central neurons are done by the oligodendrocytes. I hope this question is clear. We'll move on to the next question. Myelination in, okay, we have already discussed the answer. Myelination in neurons of the central nervous system is done by. Still, let's see if everyone is listening to the explanation of answers or not. Whoever is raising hand, can I know your query in the chat box? Okay, we have already discussed the answers. So there is no point in waiting. The correct answer is oligodendrocytes, right? So this is the central nervous system and the myelination in the central nervous system is done by the oligodendrocytes, whereas in the peripheral nervous system it is done by the Schwann cells. I guess everyone must have learned it by now. So now the eighth question is, which of these is not a neurotransmitter? Which of these is not a neurotransmitter? The options are glutamate, laminin, GABA, and dopamine. Which of these is not a neurotransmitter? Remind it, not. Its time starts now. Start answering. Who can tell me what is a neurotransmitter? What do you mean by a neurotransmitter? Can anyone define what is a neurotransmitter? No one knows what is a neurotransmitter? Transmits signals from one neuron to other. Correct. So our body can't function without neurotransmitters. They are the chemical messengers of our body, right? And we have many neurotransmitters in our body, out of which the options there is one which is not a neurotransmitter. I guess everyone is giving the correct answer. Laminin is not a neurotransmitter. But can anyone say what is laminin? Where is laminin present? Laminin is a part of which system or which component? What is laminin? Can anyone answer? We know that glutamate, GABA, and dopamine are neurotransmitters, but what is laminin? Does anyone know? This is also a very simple thing. It's a part of cell biology. Yes, yes, it is a part of the cytoskeleton. Good. Okay. So, here are the 8 types of neurotransmitters we have in our body, and each of them have different functions in our body. Adrenaline, GABA, noradrenaline, acetylcholine, dopamine, glutamate, serotonin, and endorphins. Adrenaline helps produces are produced in stressful conditions with increased heart rate and blood flow, right? It increases the heart rate and blood flow. GABA, it is the calming nerves, calms our nervous system. Noradrenaline, it affects our brain, it contracts our blood vessels and increases our blood flow. Acetylcholine is involved in our learning and memory process, thought process. Dopamine, it gives release of dopamine gives a feeling of pleasure in our body, feeling of motivation in our body. Glutamate, it is the most common neurotransmitter. It is also involved in learning and memory. Serotonin is a mood neurotransmitter. You can say it contributes to well-being and happiness. Whenever we feel happy, serotonin is secreted in our body. It also helps in sleep cycle and digestive system regulation. And endorphins are also released during exercise, excitement, etc. It is also gives a feeling of euphoria. Euphoria means excitement and as well as reduces pain. So laminin is not a neurotransmitter. We'll move on to the ninth question. The efflux of dash assists in dash membrane to resting membrane potential. The efflux of dash assists in dash membrane to resting membrane potential. Your options are potassium ions depolarizing, sodium ions depolarizing, potassium ions repolarizing, or sodium ions repolarizing. Your time starts now. Yeah, laminin is a like like a lipid like a read the question properly and then try to answer. The efflux of which ions? We have already discussed which concentration of ions is outside, which concentration of ions is inside. We'll again discuss this. May be a little confusing. That's why we'll keep on discussing this concept of membrane potential. I'll again explain you. So let's see how many of you have given the correct answer. Some are saying A, D, B, C. Everyone is confused. Like, got all kinds of answers. Which is the correct answer? Okay, we'll not waste more time. We'll see what is the correct answer. If you have given the wrong answer, it's okay. Try to learn. Okay. So the correct answer is C. The efflux of potassium ions assists in repolarizing membrane to resting membrane potential. Right. I've already discussed this answer when I was explaining the figure of the ATPase pump. I guess most of you didn't listen or didn't understand it. It's okay. We'll discuss this again. So the few steps of action potential are: first is the membrane potential, then is the depolarization, then is the repolarization, and then is the hyperpolarization. So at the resting membrane potential, our membrane potential is minus 90, and at that time only the K plus leaky channels are active, right? And when our with the opening of the, that is that time only the K plus channels are open, right? And when the, so what is happening, this concentration of K plus ions inside the cell is more, and when it is at the resting membrane potential, the K plus ions are moving out of the cell, making it negative, right? That's why the membrane potential of the cell at resting membrane potential is minus 90, and when the voltage gated sodium channels open, the membrane becomes depolarized. That is, the membrane potential starts increasing. It moves towards a positive value, right? So at when the membrane is at depolarized state, that is, there is a opening. Oh, sorry. There is a opening of the voltage gated sodium channels, and the voltage rises and crosses zero. Obviously, from minus to it becomes positive, right? It becomes to plus 35 millivolts. That is the, you can see the peak here. It is at minus 19 resting membrane potential, and it increases when it gets depolarized and crosses zero, right? It is at plus 35 millivolts, and when after it reaches the depolarized state, the voltage gated sodium channels which was open, it closes, it starts closing, and what opens next, that is the K plus voltage gate. K plus leaky channels are different and K plus voltage gated channels are different, right? K plus leaky channels are open all the time. So after the closure of the Na plus ions, what happens? There is no movement of the sodium ions, and the movement of the K plus ion starts again. So what happens? The Na plus ions are not going into the cell, and there is opening of the K plus voltage gated ions. So the membrane potential decreases again. So so opening of the voltage gated K plus channels happens, and there is the closure of the voltage gated Na plus channels. This is the repolarization event. The question was the efflux of the potassium ions assists in repolarizing membrane to resting membrane potential. So the question is, like, from repolarization, how it is coming again to resting membrane potential, right? So the K, so after repolarization, there is hyperpolarization state. When this happens, the some voltage gated K plus channels remain open even after resting membrane potential is restored. So there is a opening of the K plus voltage gated channels in the repolarized state, and after that in hyperpolarization they still remain open. So what is happening? All the K plus ions are going out, making the membrane potential negative again and reaching down to resting membrane potential. That is, the efflux of the potassium ions is happening again. It is happening continuously from the repolarized state. It is becoming hyperpolarized. That is, it is now in a much negative state. Again, it is coming back to the resting membrane potential. So, for all of these, the resting membrane potential is all based on the potassium ion concentration inside the cell. So, our answer was potassium ions and repolarizing. Okay, we'll move on to the next question. and this is the last question of our session. Velocity of conduction of action potential in muscles is dash times of that in myelinated nerves. So there is this action potential occurs. We understood what is action potential, the resting membrane potential, depolarization, repolarization, hyperpolarization. But this process, it occurs at a speed, right? Like the steps, like the occurrence of these steps, what is the velocity of the occurrence of these steps? It is differing from regions to regions. It is happening at a different speed in muscles and it is happening at a different speed in nerves. So that is the question, that the velocity of the conduction of action potential in muscles is dash times of that in myelinated nerves. I hope everyone is have understood the question. I was trying to explain the question if anyone is getting confused. The events, that is resting membrane potential, depolarization, repolarization, and again hyperpolarization, all of these events are occurring in all the cell membranes of the cells, but it is occurring at a different speed in different regions of the body. Like in muscles, these events occur at a different speed. In brain, these events occur at a different speed. So it is asking the muscle velocity of action potential is how many times of that in myelinated nerves? 13, 1 by 13, 1 by 2 or 2? Everyone is writing D or C. I guess most of you are not attending the lectures. You need to go back. If you're giving the wrong answers, it's totally okay. You are learning the correct answer through this session. But if you're giving wrong answers, please go back and listen to the lectures again. Surely you will clear your concepts more elaborately, right? As we are time restricted here, I am unable to explain you the concepts of all the questions in a very broad manner starting from the basics. You can learn that through the classes, right? Okay, I guess most of you have given your answers, but unfortunately most of you have given the wrong answer. I could not see a right answer here. The right answer is B, 1 by 13. The velocity of action potential in muscles is very, very less than that of the myelinated nerves. We will see here. So the muscle velocity, this is the muscle fiber conductance velocity, is the velocity at which muscle fibers transmit action potentials prior to muscle contraction, right? And this muscle velocity is susceptible to the environment condition, the type of muscle fiber, the measurement of muscles. All of this depends and it varies from muscles to muscles in healthy individuals, but approximately the muscle velocity, the velocity unit is what? Miles. Here we are seeing in meter per second. So it is 3 to 5 meters per second. Okay. This is the velocity of muscles. Whereas in myelinated axons, these have the fastest signals in our body, that is 80 to 120 meters per second. That is, they run at a speed of 179 to 268 miles per hour. It is very fast. These are the fastest signals, but muscle signaling is very slow. Why the signaling of myelinated axons is faster? Because of the saltatory conduction. What is saltatory conduction? These steps occur like you run or you jump. There is a difference, right? You run from one position to another and you jump from one position to another. So saltatory conduction is that in the, and you can see here, this is the myelinated neurons, this is the myelin sheet, and these are the nodes of Ranvier in it. So when the channels open, there are plenty of positive ions inside the cell, you can see, right? And it almost looks like the signals are jumping from node to node. So in the myelinated shields, because of the presence of these nodes of Ranvier, where the action potential or the movement of ions is taking place, It is like happening like from node to node, and it is jumping from one node to another node in a process called a saltatory conduction. That is why the signaling is very very fast. You can see they are moving by leaps or jumps. That is why the action potential in the myelinated axons is fast. It is not with unmyelinated axons, but only myelinated axons, because they have nodes of Ranvier in it. Okay, I guess everyone has understood the answer. We are at the end of our discussion. I want to know your scores out of 10. How much you have scored out of 10? Answer honestly. No one wants to answer. Is the score too bad? Okay, the neuron potential, the resting potential may be different for neurons and muscles, but we are not talking about that. We are talking about the velocity of conduction. At what velocity this process is happening in muscles and neurons. Okay, so that is different. That is the 1/13th of the speed of the neurons is present in the muscles. Like exactly not 1/13th, but in an approximate range. It is not obviously half. We can see from the values here. We're talking about the velocity of conduction. Okay, 7 by, I could see only 4, 5 answers. Rest all of you have not counted your answers. Okay, 8 upon 10 is a good score. 7 is also good. But if you attend the lectures and then come, obviously, no, no, no. Do velocity and action potential mean the same? Definitely not. Action potential is the movement of ions across the cell, right? Sodium and potassium ions that we are discussing here. But velocity is the speed of the process. This action potential is divided into four parts, right? We discussed resting membrane potential. It is at a resting phase, then it gets depolarized, then it gets repolarized, then it gets hyperpolarized. These steps, at what speed these steps are occurring in different cells. In muscle cells, the speed is different. In neuronal cells, it is different. And myelinated axons, it is happening at a very fast speed. Okay. Okay, it's already time. We'll disperse here. We'll meet again in the next class. I request everyone to please attend the lectures and then come to the class. We'll have a better interactive session. Okay, and if you have any queries regarding this session, please write. You can write down in the discussion forum at the NPTEL class, NPTEL website. I can check that. And also, if you have missed any of the portion and if you want to revise this session, I will be posting these recordings in the YouTube channel. You can watch it there as well. Okay? Okay. Thank you. Thank you, everyone.